And now, kindly, I would like to present Robert Taylor from Malvern Panalytical. Actually, I'm giving you the host permission to share data. Hi, John. Hi.
Hi, Shlomit.
Hi there, Shlomit.
I will appreciate it if you could open the camera, guys. I can see your presentation already.
Yeah.
Go. How are you doing?
Good, thank you.
Okay.
Hang on, I'll try to get my camera on.
Okay. Good. So we are about to talk about spray analysis.
Okay. Just see me okay?
Can you make it presentation mode?
Yeah.
It's
Okay.
good. Thank you for arriving. Thank you for open camera. It is nice for me to see you guys.
That's fine.
Let's start.
That's fine.
Well, yeah, and thank you, Shlomit, for the introduction and giving us the opportunity to talk here as well. So, I mean, on that, I'll be doing most of the talk today, but luckily my colleague, Dr. John Ddungu, is here to support with any kind of questions that we get, particularly with the spray analysis side of things. And that's what we're going to focus on today. So we're looking at the development and bioequivalence of OINDPs using spray analysis and another technique of automated microscopy. I've got a number of slides here, and I think I've probably got a few too many for the time that I have available. So I'm going to probably go through them at quite a pace and keep to the salient points, with the hope that we can get all the way through to the end here and start having a nice discussion. So I mean, what's really valuable for us and for many of the people on here, I imagine, is to have a nice discussion around this topics and understand what your experiences are. So if we can have some time for that, if we do receive questions, then more's the better, I think. Okay, so just very briefly, this is sort of a quick run through of what we're talking about here. So, I mean, OINDPs, they're termed as complex drug products, and there are several definitions of complex drug products. But the two that I'm going to focus on today is really looking at the route of delivery and the actual drug device combination, because as you can see from the graphic at the bottom of the screen, OINDPs are a combination of the formulation and the device. But this first one here that I'm discussing about route of delivery, most of these drug products are intended to have a locally acting effect. So, you know, we hope to sort of deliver the active ingredient to the nasal or lung epithelia and treat conditions there, usually some kind of inflammation. Some products, of course, are intended to get into the systemic circulation, but most aren't. And that means it's quite difficult to understand what the performance is of these locally acting drug products. I mean, how can we determine whether you're getting a good performance or a bad performance, or indeed is it the same or different to the innovative product if you're making a generic, for example? And then similarly, the drug device thing, well, the actual performance is not in the formulation alone, it is how that is aspirated through the device itself. So this is another complexity that is added onto these types of products. All right, and then this is sort of some comments from US FDA. This is something that the former commissioner, Scott Gottlieb, had mentioned when he left back in 2019. He just comments on the same kind of things, really. I mean, these things are complex, and due to the nature of their formulation, they are hard to understand or to sort of measure their performance, and therefore hard to copy. So making generics of these things is quite difficult, and part of that is because they act locally. So this local sort of local acting feature of these types of products means it's difficult to sort of quantify and qualify how well these things are performing, and therefore getting your product through the regulator and determining that it is in fact bioequivalent is one of the challenges here. But The good news is that the FDA are fully supporting trying to promote new tests, new in vitro tests, new analytical tools to help speed up that process. And this is really where we think Malvern Panalytical can really aid in this situation. Okay, it all kind of comes down to these sort of three metrics. That, again, this is kind of US FDA focus, but I mean, they highlight three areas of bioequivalence. Qualitatively the same, Q1, quantitatively the same, Q2. These are relatively straightforward, just making sure you have the same API, same excipients, same dosage, that kind of thing. But then this third one here is about something about the physicochemical attributes of the active ingredients, and the reason being is that if they have the same physical chemical attributes, they're likely to perform the same. Okay, and that's really what this tetrahedron characterization tetrahedron is describing here. So if you know something about the properties, you measure something about the structure, these are intimately related to how easily you can make something, and then also how that performs in vivo. Okay, right, first technology, spray tech. So what you can see on screen here is, and I'll go through this in a little bit more detail as well. Effectively, a laser is shining through a beam path here. We have, in this particular case, a DPI aspirator. We have a USP throat, and you aspirate it through here, comes down through here, and this is where you can measure the particle size distribution of the spray plume that emits from the DPI, and then you have this cascade impactor there. Okay, and so what you do is you attach a vacuum pump on the end there, so you have different aspiration rates as well. Okay, so the way this works is that we have particle size distributions based upon the laser diffraction technique. And for those of you who are not so familiar with it, essentially you have a laser. So in other words, you have a monochromatic beam, which is coherent, impinging upon some matter. Okay, and then the scattered photons from this matter then can interfere with each other constructively or destructively to produce what you see here, these kind of lobes of high and low intensity. Okay, and so the position and intensity of these lobes can then be related back to the particle size distribution of the material itself. So all you do is you take a cross section of this, determine the angle at which these lobes are emerging and the intensity, and you can infer the particle size distribution. Okay, so one of the nice features about laser diffraction, it has a very wide measurement range. So for spray tech, we're talking 100 nanometers to 2 millimeters. That's quite substantial. And it's actually probably a bit overkill for what we need here, because you know, we're talking inhalable fractions are between sort of 1 and 10 microns in size. Those in the nasal spray region are probably tens of microns higher than that. So well within the range of what we're interested here. But also it's rapid, and this is really important because the spray plumes, they will develop and emerge and then dissipate very quickly, less than a second it takes. So you need to have rapid data collection to actually understand what's happening to the spray plumes themselves. Okay, and so we have two arrangements here. One is looking at a nasal spray actuator. You can probably see it just there. So we have just an actuator to pump something into the laser path, and then I just described this sort of DPI impactor type setup there as well. Okay, and then here's a bit more of a detailed schematic of the spray tech. I won't go into too much detail here, but effectively laser shines this way, goes that way, and then you have spray plume emerging here for particle size distribution testing. The next technology and techniques, I guess, is automated image analysis and something called morphologically directed Raman spectroscopy. So I'll describe that in a second now. Here's the technology. This is the Morphology 4 and the Morphology 4AD. Effectively, it's a light microscope with automated XYZ stage and means to disperse your sample. So the idea being is that you can automate the whole process of dispersing the sample, measuring its particle size and shape, and with the integrated Raman spectroscopy, you can then also determine the chemical identity of these particles as well. So useful for those products which have which have multiple components in them. Right, so what we're doing here then is that we're taking images of particles, determining their size, determining their shape, and because we're measuring many thousands of them, we can get some statistical information, so distributions. This one's of a size distribution, but of course we can also get some shape distributions as well. And the ultimate goal here is to make some inferences about your sample. Is it mostly single primary particles, or do we have agglomerates present? Are they regular in shape or irregular or kind of elongated like this one? Rough, smooth, dark, light, that kind of thing. And then because we have the Raman spectrometer integrated into this system as well, we can also start to understand, well, what's the chemical makeup of these particles? And if you have multiple components in your product, then you can then separate them not only on their particle size and shape, but also on their chemical identity. Right, so that's that in a nutshell. On to the first case study, and this is on nasal sprays, looking at plume. So we're looking at the sort of the spray type technique here. And there's some guidance for industries we see here. So we have some regulatory guidance. This is actually quite an old document, but it still holds true. And some recommendations for this Q3 type characteristic. So we're looking at sort of the physicochemical properties here. They're all listed there, but the two that I've highlighted here are the ones that are relevant to this application. So droplet size distribution, so that's what we're doing with spray tech, and then drug particle size distribution, that's what we can do with the MDRS technique. Okay, so this is the expected data output from spray tech, and effectively what you have is size on the y-axis versus time along the x. You can see there that from the key that we have the D10, D50, D90. So hopefully you're familiar with sort of particle size distribution metrics. Okay, and then the time, as you can see there, we're only going up to sort of less than 200 milliseconds. So the whole spray process, the plume development process, is less than a couple of hundred milliseconds. And then what we also have here as well is the transmission, and we'll go into why that's relevant later. But effectively, if there's a higher concentration, then the transmission drops. So we can start to infer something about the content of the spray as well, or the mass that's being emitted. Okay, but something of interest here as well. So let's just talk about this just very briefly. But we can kind of divide this whole spray pat up into three stages. The first stage would be the formation phase. This is, I guess, happening right at the beginning of the actuation. And then we get into this fully developed phase, where you can see from these time profiles how stable it is. And this is really this kind of stable region here, where you're going to get most control out of your device, okay? And then finally, as it dissipates through, you get this final stage here where the particles begin to coalesce and they just get bigger and you lose that control, okay? But we can measure the particle size distributions of each of these stages. So you have the formation phase there. You can see the D10, D50, D90, you have the fully developed phase and then the dissipation phase as well. In this case, we can display this as an undersized chart. So if you're familiar with this, it's kind of a cumulative frequency curve. So the first 10%, that relates to the D10. The 50% relates to the D50, and then the 90th percent relates to the D90, okay? But you can see from these figures here that the formation phase in blue, it starts out quite coarse, all right? And then as we get into the fully developed phase, it shifts left. So we get smaller particle sizes. And then finally, as it goes to the dissipation phase, it starts to coalesce and get bigger again. But why this is relevant then, if you relate it back to what this is, the structure, and why this is useful, I mean, you can see these particle sizes here. Okay, so this being a nasal spray, what you don't want to do is have your particle size too fine, because if it is too fine, then it could be that some of this drug products will get into the lungs, and that's perhaps not the desired area for it to be. Could have a toxic effect, in fact. And then also, you probably don't want it to be too coarse either. And the reason being is that we want to have a pretty good coverage of this product, across all the nasal epithelia in the nasal cavity. So if it's too coarse, then maybe you'll get isolated spots of this product depositing on sort of localized areas within the nasal cavity. And if anybody here has used nasal sprays kind of at the end of their life and have kind of misused them somewhat and got quite coarse particles, sometimes it can be quite irritable or irritating. So making sure you have the right plume particle size is really sort of going to control that performance element of the drug product. Okay, so that's the kind of data that we're looking for, and in this example here, we're actually looking at different, changing the viscosity of that formulation by increasing the loading of the excipient PVP. So there you go, increase the loading, increases the viscosity, and you can see from these time profiles here, by doing so, you go from a relatively small particle size of the D50, that is, in the fully developed phase. We just having water. Okay, that's water down there. As you increase the loading, the particle size increases. That's probably expected. We have higher viscosity, etc. But get beyond 0.5%, and we're now into this very coarse, very uncontrolled particle size distribution. All right, and that's the undesirable thing. We want to avoid that. What's interesting as well, I think we will see this on the presentation that follows this from Proveris. This is a nice sort of description of the plume geometry, and you can see there at 0.25% PVP, we have a nice wide angle of the plume geometry, so we're expecting a good spray pattern, good coverage of the nasal cavity. And now compare that to the 1.5% PVP. These are now very coarse particles. Lots of inertia. These are just going to jet straight out of the device, and again probably deposit at sort of inhomogeneous or rather localized areas within the nasal cavity. So again, not quite so good for the patient. And then we could also have a look at different devices. So this is a device where you can load up the pressure and control the actuation. So actually we get a very nice prolonged developed phase using this device. That's pretty good. But again, we see that as we increase the loading of the PVP, we increase the overall particle size. So again, these are the D50 values, and it gets to these quite coarse and somewhat uncontrolled particle size distributions when we get above 1% PVP. So interesting to see there that you can use different devices too. And then finally, you can do some funky things with rheology. So what we've got here on the right-hand side, you can't see what sort of shear rates are used here, but in the beginning here we have very low shear rates, and this means that we have quite a high viscosity formulation in this case, which is quite valuable for these types of products in terms of stability. So if you're looking for longer shelf life, then perhaps you want to have high viscosity formulations when it's sat on the shelf. But as you increase the shear, so when you're actuating the device itself, you're breaking up a lot of the microstructure within that formulation, and the viscosity drops away. And as we've seen from the previous slides, low viscosity is beneficial for controlling that plume particle size. And then finally, as the actuation finishes, the shear rate goes to low levels again, and the viscosity also returns to what it was before. So this is kind of using shear thinning type rheology technology to optimize the stability of certain products. And again, from these time profiles, this is where we're actually actuating at different speeds. So we have 40 millimeters per second, 70 and 100 millimeters per second. And we can see that at these two higher speeds of actuation, we get a pretty nice looking developed phase. We get down to low particle sizes, that's good. But at 40 millimeters per second, it's bad. It doesn't really give quite the right particle size. It's a bit too coarse. And also it highlights a problem here with respect to how users actually use a device itself. So if they're struggling with dexterity, if they're not able to actually actuate the device fast enough, they might not get the right delivery. And that's a key thing to understand when you're developing these products. Okay, Next case study, different technologies, same products and our nasal sprays, but looking at the drug particle size distribution. This is with using the MDRS technique. So we're using automated image analysis coupled with Raman spectroscopy. And what we have here is a nasal spray suspension, and we're looking to do an imaging scan, pick up the particles, and then analyze them for their chemical makeup. And then we can determine what the particle size distributions are of the two components. Right, so here we go. We've done a scan and we've got some particle images, and then we've also collected some Raman data. And what we can say is that some of these particles are of the active ingredients, like this one, and then some of these particles are of the excipient. But what's interesting is that if you eyeball these two ones, you can quite clearly see this one is elongated and this one is a bit more spherical, okay? And when you look at this in terms of the whole population, you can actually see the API is indeed on the elongation scale is actually quite low. So these are more spherical in shape, but the excipient has a higher level of elongation, and these are more kind of needle-like in their appearance. So what we can do here is we can apply a filter, remove all this stuff here, which we know is not of interest, and focus on this material here, which is the active ingredient. Okay, and this is how we're going to develop bioequivalence in this case. Okay, so we've run two scans, one on generic, one on the reference listed drug. We've isolated the active ingredient through the MDRS technique, and now we've compared the size distribution of just the active ingredients alone. And this is why we can demonstrate that we have bioequivalence between these two products. Okay, but if you're only able to sort of capture the whole all the particle sizes that are present, then you wouldn't necessarily get bioequivalence there, because the vast majority of those particles will be excipient. Okay, so that's the power of the MDRS technique, to isolate what is relatively low concentrations of active ingredient and then compare that to between two different products. And then just a word from the FDA again. This is sort of leading back to what was said by Scott Gottlieb in the beginning of the talk about how the US FDA is trying to promote these new technologies, and this is one such case that they refer to. And just as a final point here, in the product-specific guidances of similar products, it also says that morphologically directed Raman spectroscopy can be used as a means to generate Q3 bioequivalence. Right, how are we doing for time? Yeah, not too bad. So on to the next one, dry powder inhalers this time, and still we're looking at inhaler plumes, okay? But if you look at the guidance now, it's quite different to nasal sprays, because what we're looking at here is really what's the content of the actuation from the DPI device. So they typically are interested in what the compactor profiles are showing us, and also at different flow rates too. So looking at 30, 60, and 90 liters per minute. What's the single actuation content, and also what's the aerodynamic particle size? So where do these particles impact within, say, a cascade, or latterly a next generation impactor? So that's what the regulatory guidance is. But what we can do is we can support those activities. So with laser diffraction and MDRS, if there are, for whatever reason, some differences or some unexpected results coming through from your impactor analysis, we can use these techniques to help develop your formulations, or indeed to help deformulate innovative products for a generic approach. But the same principles apply. Okay, we have, in this case, this is the AstraZeneca's turbo inhaler. This is kind of the actuation device here. We can meter the dose, and then we have air flowing through, which entrains the dry powder. We have a means to disperse it up here, and so we're going to be able to measure the particle size that is emanating from this end here. All right, so more spray analysis now, and again you can see we have size versus time series here. We have two formulations, A and B, And what you can immediately see there is that formulation A has across the actuation, it has a much sort of larger particle size. So it starts off large, and then towards the end, it then drops away. Okay, but formulation B, on the other hand, starts off small, gets large, and then finishes off small again. And this is also reflected in the particle size distributions here. So again, you can see formulation B has a much more pronounced fine fraction. But again, it's also worth pointing out that, you know, what is it that you actually need for this device? What's the intended purpose? And well, clearly this coarse fraction here is much too large. It's never going to get into the lung. These particles will impact far too high up in say the trachea or perhaps in the bronchi, but they're never going to get in through to the deep lung at this particle size. You need something at this end here. So this is how we see this technology being used. If you adjust your formulation to promote the fine fraction, that's what you're interested in. That's what you want to develop. Okay. And then similarly, we have two different formulations now, and what we're looking at here is the effect of the different flow rates. So again, we've got 30, 60, 90 here. And well, you can see there that with 90 litres per minute, so at the fastest flow rate there, we're getting most of the material coming through as the fine fraction. So 1 to 10 microns in size. This is ideal for DPIs, but you also have this coarse fraction here as well, which will just impact too high up and won't have any sort of effect on the disease state. But if you go down to the lower flow rates of 60 and 30, you can now start to see this coarse fraction emerge. And of course, that's not good for the reasons I've just described, but it also points out that, again, there's going to be some user compliance issues here. If you have, in order to get the best out of this product, you need to ensure that you have a good, fast inhaled breath to make sure that you have all the fine fraction there. If for some reason, or for whatever reason, that you can't inhale very strongly, then you're not going to get the full benefits of this product. And obviously, that's not a good thing, because it will lead to a heck of a lot of variability across a cohort of patients. You know, some will inhale fast, others will inhale slow, and this will lead to significant differences. So reformulate this, put some mag stearate in. Mag stearate is a known lubricant used for sort of peroral products, but it's also good for kind of keeping particles apart. And this is why we now get much more consistent particle size distributions at the full range of flow rates there. So by adding in or changing the formulation slightly, we've optimized the particle size distribution, and therefore we would expect a reduced variability across the patient cohort. Okay, and then finally, just on this topic, I mentioned at the beginning of the spray analysis technique, you also get the transmission data too, which gives you some information as to the actual content of the spray. And so again, the lower the transmission of light, then it indicates the higher the content of the spray. And again, we can see here that at different sort of flow rates, we get slightly different content. So if we convert that transmission data into concentration, so ppm here, we can see at 30 litres per minute, the concentration is fairly low. It gets up a little bit to maybe 6 or 7 parts per million, but then tails away. But if you have the faster flow rates at 60 and 90 litres per minute, then we start to get a much higher concentrations, and perhaps this is where we want to be. So in terms of actually delivering the full dose, again, we need to have a relatively fast flow rate to get that. And again, that brings questions about patient compliance. Okay, so again, this is just ways in which we can use this technology to help develop such products. And well, if there was an innovative product that had this kind of behavior, then this is what you would need to match it to if you're creating a generic. Okay, so going back onto the MDRS technique now, again on DPIs, and this is actually looking at the performance of next generation impactor. So for those of you who are not familiar with an NGI, essentially you aspirate the device through this. There's a vacuum being pulled, and the plume gets pulled through these various stages, and you can see that there's different size holes there for which the particles can flow through. So the principle being, the higher they impact, the larger the particle size, and therefore if they impact at sort of the later stages, then the smaller the particle size. And this will give some information as to where you expect these particles to deposit in vivo. Right, so here is the output from an NGI test. It's basically quantifying how much drug is deposited at each of the stages in the NGI using LC, liquid chromatography. Right, so in this case, we're looking at the active ingredient fluticasone propionate, and we've got two DPIs, two products, and ultimately we can see that we have significant differences of this deposition profile. Okay, particularly if you ignore the pre-separator here. If you're looking now at kind of the later stages where we expect them to go into the lungs, then differences really kind of emerge at stage 2 and at stage 4. So DPI 2, we can say we get a lot of this FP depositing at stage 2 compared to DPI 1, but at stage 4 it's the other way around. So what we can say is that DPI 1 is probably performing slightly better, will probably give a much more effective dose because it's getting further into the lung. This is what this is inferring. Right, but why is this? Why do we have these differences? Okay, here's the setup. We have this kind of modified NGI cup. That's what you see up there. Again, inside that little insert fits this quartz disc. Okay, so you put this quartz disc in there, that goes in the NGI, and then you collect the particles that deposit on those discs. What happens now is that you scan certain areas of these discs to determine the particle size and shape distributions of those deposits to try to see what's going on. So here we have then some size distributions. This is of stage 2, and you can see there that the majority of this is in the 1 to 10 region. That's fine, that's expected. But also we have this rather large peak of coarser material, and in fact this is known to be lactose. So this product has 2 types of lactose, fine and coarse. This is the coarse fraction coming out here. Okay, but ultimately this is both the API and the excipient all wrapped into one. Okay, and that's stage 2. Stage 4, we've now lost that coarse fraction there, and that's because it's just simply not fine enough to get through to these higher stages. Okay, and so what we see now is everything is fitting nicely, a nice kind of monomode distribution in between that inhalable fraction of 1 to 10 micron, and this is the size distribution that we get. So okay, that's what we get. But what you can see from these images here is that some of these particles look different. So what are they? That's where the Raman comes in. Okay, so what we see on screen now is a series of reference spectra, and these first 3 at the bottom relate to the pure material. So we have API 2 in red, API 1 in blue, and then the excipients in black. Okay, we're only going to focus on one API, one of the APIs in this particular study. But you can then therefore have combinations of these 3, and that's what you see in this top 4 spectra. So you have combinations of APIs 1 and 2. You have combinations of API 1 and excipient, 2 and excipient, and then all 3 together. Okay, but the idea being is that should you then get a spectrum, which is one of these kind of hybrid spectra, you can say that that particle was a multi-component agglomerate of 2 or more particles, and this is how we determine what they are. Right, so there's quite a lot of information on this slide, but what we've done then is that we've run the scan, we have collected the Raman data, and we have determined how many particles are of the active FP, how many particles are of the lactose there, and then how many particles are this multi-component agglomerate between the FP and lactose. And this relates to stage 2, and then this relates to stage 1. Okay, and you can see there that as a percentage of total volume is something like between 8 and 15% of both the DPI products is the free FP, and then we have something between 50 and 65% is the free lactose, and then between 20 and 40% is this multi-component agglomerate of FP and lactose. But of course, what it's also showing us here is that if you look at DPI1, there's a lot more free FP, but there's also a lot less of this multi-component agglomerate, or rather there's a lot more in DPI2. And now if you consider the multi-component agglomerates will intrinsically be bigger than their primary particle constituents, these are going to be rather large particles, large aerodynamic particle size, and these will impact higher up in the NGI. Okay, and so what's interesting then is that DPI2 is kind of confirming what we've seen from the NGI data, okay, which is that more FP is impacting here because it's agglomerated to lactose. Okay, and now if you look at the stage 4 here, we get a similar kind of pattern there. So we have lots of free FP in DPI1, a bit less for DPI2. And then again, also we have a little bit more of the agglomerated FP for DPI2. So it's kind of telling the same story, that ultimately the difference between these two DPIs is there's more free FP in DPI1, and there's more agglomerated FP in DPI2, and that's the reason for the different deposition profiles. Okay, so that's how we can use this technology to either deformulate. In this case, we're comparing innovator and generic, so that's a deformulation sort of activity, or indeed if you wanted to optimize your formulation candidate, then you can use this technology for that too. So again, this just summarizes what we just said there. 2 DPI products. The second DPI product had the active ingredient impacting further up. This was related to the prevalence for FP to form a multi-component agglomerate, and that these are bigger in size, and that explains why the impact higher up, and hence more FP will impact at earlier stages for DPI2. Okay. Right, so how are we doing for time? Oh, 4 minutes left. Right, yes, another.
Hi, Robert, how are you
Yes.
doing? It seems like you have a lot of data to share. Can we order another session?
Yes, well, if we have questions, I can take them now, or we can just forego this last application.
Okay, so I'm gonna ask my customers, guys, ladies, do you have any question for Robert or John? Because Ian from US already woke up very early for us. Already with us. Questions? No, I cannot see any question regarding spray analysis in Malvern. But Malvern Panalytical, you're marvelous. You're wow. Just great data and great guys. These guys are doctors. True? Yes, you are. Thank you, guys.
Okay.
Robert, I'm gonna order another one. Oh, we have question actually. What is your experience regarding eye drops, SUSP, or whatever?
Can you repeat the question, please?
I'm gonna let Yossi ask you directly. Just a moment. Yossi, I'm gonna unmute you again.
Hello?
Hi.
Okay, now I see that you can hear me. In eye drops, do you have any experience regarding eye drops where you contain a few APIs, not only one? And I'm speaking about the active pharmaceutical ingredient and the anti-inflammatory, and which did you ever test the PVP qualities or the concentration that should be used?
Not personally, no. I mean, this is it's actually information that we would like to have as well, because I mean, whilst we supply a lot of this technology and we support many of our customers in their development activities, they tend to keep the rest of the details to themselves. In terms of eye drops themselves, I've seen 1 or 2 applications of them, yes. In terms of whether you can get multiple APIs, so a combination type product there, yes, we've done that as well. I mean, as long as they are sort of chemically distinct from one another, you can use the Raman spectroscopy to isolate one from the other. So in fact, that DPI example did have 2 APIs in it. So I mean, hopefully that answers your question, but I can't recall any particular case study where we've looked at the effect of excipient in eye drops. But my guess would be is that, again, if it's impacting things like viscosity, then how's that going to affect the distribution of the eye drop across the eyeball? Is it going to spread well, or is it going to remain isolated where at the point where it's administered?
Yeah, you have some kind of steroids and some kind of API that is doing some pharmaceutical action in the eye, and then you have some dispersant like the PVP or coater or whatever, and both of them should comply with the particle size distribution.
Yeah, I mean, absolutely. I mean, again, I haven't seen too many eye drop formulations. I'd imagine that if you wanted to have an eye drop product that gives a kind of a sustained treatment, then you probably would want to have a suspension in there, which can slowly release the active over a longer period of time, assuming that doesn't create some kind of irritation. But then if you wanted a much quicker impact, an immediate release, let's say, then possibly a solution would be better, and therefore particle size isn't really of an impact in that case.
Thank you.
Thank you, Yossi.